Welcome to video one in our course B09 R for AI. These are introductory videos to this area. So there's quite a lot of reference material that could be used, but my suggestion for learning effectively for beginners, which is the target of this course, is to use tidy modeling with R by Max Kuhn and Julia Silge. Um, that's a very effective group um, uh, s- resource where we're mainly focusing on the first component rather than the more advanced second component. There's also a large amount of in material available on different resources on fair data, which is the focus of the first part of our talk, because if we want to use AI methods, we need to have good quality data. If we have poor data, it's kind of pointless trying to use AI methods. So our goals for this course are to learn about AI data and the associated methods and be able to model data with R. In some senses, AI methods are just a small component of statistical computing. And we'll also try to apply a very, very simple supervised learning model in R. So that's kind of our focus here. So think about AI ready data, AI ready with a Y would help. Think about linear regression as a simple statistical technique, and then apply a form of linear regression in R. That means by the end of this course, you'll be able to consider different AI methods and the data required to assess them, apply linear regression and similar techniques to high-dimensional data, and then also perform supervised learning in R. That's the goals of this course, which again, I reiterate, is aimed at beginners. So let's think about the topics on this course. We're going to first focus on making your data AI ready and think about that being a key component of that before we think about further about AI methods, getting data and exploratory data analysis, which is our first protocol before we do anything else. Then we'll think about linear regression, and then we'll think about using supervised learning using R. So that's our approach. So let's first think about the complexities of modeling. Um, that if we're thinking about a model selection and running a model and optimizing it, there's a lot of choices and you need to justify them. And you consider, and that's what we'll consider first in this course, is what raw data are you using? Do you have enough data? Why are you using this data set? What approach you're taking in terms of your statistical modeling? Why is that the best approach or the one that's most commonly used? Would you get the same results with a different model? And then if you're making predictions or classifications, in what way are they relevant for others? And again, a question to ask yourself, if you're doing this in 5 years' time, would you be doing it differently, irrespective of changes in technology and other things? And this links to research integrity, rather than just shoving your data through a black box where you want to understand what's actually going on inside. So the question then is when to use AI. So if you've large, high dimensional complex data, machine learning and similar approaches can be effective for that where you want to infer complex patterns, make predictions, or automate decisions. There's can be a strong predictive approach for predicting continuous or discrete data using AI-based tools, depending on what you're trying to do, especially if there's copious time series or historical data. And if you need a system that can uh, update itself in a sort of Bayesian framework with new data, that can also be effective. And that's very good for complex biological systems where the data available changes over time because the dynamics of the system are changing over time. A classic example of where we can use AI-based methods or where we have something where we have an example would be a set of words where we have different people that are indicating, illustrating these words, and we have very similar patterns, but for a person, for example, that can't read sign language, it might be difficult to distill what is going on. And therefore, if we think about translating um, words into sign language and back from sign language to words, that's where we can have a lot of data. We want an element of accuracy. We intrinsically have an element of inaccuracy. Um, and that's the same as if we're translating languages from Irish to English or something like that as well. There's lots of examples in which ways in which we can think about AI and classification tools. We think about which is a muffin and which is a small dog. There's lots and lots of examples of these where you can think about the image what our capacity to detect things. More relevant to biology, we can think about detecting uh, difficult to identify virus particles in amongst a series of cells where the um, image represented can be quite different from each other and sometimes very, very hard to resolve fully the extent to which the types of virus particles present and to identify how many are present. And one other comment is that using AI should not be a default. If you've small data or one that is quite biased, imbalanced, it may not be relevant to do that. Um, because you need to know why a model is producing a particular outcome. If we can't find the information about the mechanism behind the differentiation between groups A and groups B, biologically, that is often not useful. Um, if you find the associations within your data set are weak, using other methods are not going to make them magically stronger. That's just the reality. You will need high performance computer access for anything that's an advanced model. And in simple terms, if you're trying to light a candle, you don't need a flamethrower. And sometimes simple methods are your most effective approach and the ones that are most easily interpreted as well. So let's think about our steps here. We might have data that we collect. We want to manage this data, organize it, curate it in the right way so that we can clean it effectively and then we're going to talk about this more about how to clean our data effectively because we'll need to transform it or rescale it in some shape or form to model it. We can do exploratory data analysis to explore what features it might have in terms of the correlations between different variables. And we may also want to tune our model depending if we're choosing different models um, and select our models and then refine our approach in some sort of way. And this is often an iterative approach that we may clean our data, but then after modeling, we have to clean it again and repeat this a few times till we find we have the right set of parameters, models, and samples that are relevant for our question at hand. And this can take some time, especially the data cleaning component. And this is a nice illustration from the R4 data science resource, where we import our data, we tidy it up, convert it into tidy format, we transform it in some way, visualize it, model it, and then continue this process several times before we're really able to interpret it. So the challenge really for a lot of large scale data sets is this point where we're trying to interpret it. This part can be often routine and not too difficult, but the key is interpretation, and um, that is often the bottleneck. And again, we can think about data science and cooking, and this parallels here that we have certain ingredients, we have certain algorithms and models that are available, and certain outputs at the end. And if we have very similar but non-identical models and similar but not identical ingredients and so on, we will get slightly different dishes at the end. Having a perfect dish is maybe not always what we want. We want to avoid bad dishes and make sure that we have ones that are of a reasonable quality. So that's just an analogy for this whole area. So why does making your data AI ready matter? Because there's substantial issues over irreproducible research. So there's selective reporting, pressure to publish, poor statistical power or poor analysis, um, lack of replication within a lab, insufficient checking by people with more experience. The methods are encoded and available, poor experimental design, or the raw data is not available. And these kind of all mean that we can't reproduce our research. And if we're trying to use advanced methods on complex data sets, if we can't reproduce it, then it may not have any value at all. And in fact, if we can't, if that is the case, then it's really not useful. So this can rise to many areas. For example, pressure from senior staff on junior staff to produce particular results. Small sample sizes are a major problem in biological research that we may have too few samples available for logistical reasons to actually address the question. So if we can't address the question properly, then maybe we shouldn't try to do that. P-hacking is a tendency of people to run, these statistical tools that run a whole pile of tests on data, and then they cherry pick the ones with small p-values. Harking is hypothesizing after the results are known. So that can lead to spurious associations between two observations from the same data set that may be independent. And setting up an experiment to test that dependency has to be inbuilt into that data. And then there's also reality is that we all make mistakes. And there's substantial literature, for example, looking at the tendency of Microsoft Excel to mangle gene names and the consequences that in research that we can see that that is also an issue. So, have you failed to produce an experiment? Many have done so. Someone else's or my own. You can see across chemistry and biology that this is a major issue. And in this nature survey, many consider that there's a significant crisis in terms of reproducibility. So why is this challenging? Because your data sets that you're given often are large and diffuse. If you have new technologies, you get new data types all the time. The reality is like storage, managing, and organizing data is expensive. If you shared resources, you get the tragedy of the commons where, in simple terms, people pee in the pool, pee in the swimming pool. Um, Often, especially if you're a newer researcher, you may find that you're given data that are already disorganized, and you're relying on data sets that predate your arrivals. You may be given old results, but you don't know how they're generated. And you're also unclear what you're going to be doing and what data you'll use. So, my term for this your arrival in this area so that you're already in data debt and you're like this monkey here. You're a bit confused about what is going on and take some time to actually figure this out. So you can think about ways in which we can try to minimize this or reduce this where possible. And if you find yourself thinking on Friday evening that you'll finish this on Monday and you come in on Monday morning and you're like this, you'll know that having good data management is important. um, and you also see this in papers. I don't know if this image came out properly, but you also see this here where data will be available on reasonable request, which is basically not shouldn't be tolerated in the current era at all. This turn. Another example is this is the realities of being a scientist, that we think, oh, we have very complex data, that we're in this situation where you have complex data and we need some sort of magical tools to tease it apart, whereas in reality, we need often very simple tools that will tell us a lot from our data. But getting the data is the biggest challenge, and getting it organized into a format that will make it accessible for analysis is the bigger challenge. And actually applying some stats to it is not difficult at all. So to achieve this, we should try to make our data findable, accessible, interoperable, and reusable. FAIR is the acronym. We may be able to make it findable. We might make it more challenging to make it accessible and interoperable and reusable. But we're trying to go along these steps to at least achieve some of these things where possible. We may not achieve all of them. Making things easily reusable is something that can be true for a certain period of time, but not necessarily for the rest of human history. Um, and this is widely acknowledged as being tricky and requires real effort. For example, we can make our data, so we might have primary data or secondary data, but we can link it to its metadata, make sure it has a unique identifier, some sort of code, and it's in a database. If it's not in a database, then it can be in a secondary database like Zenodo or Figshare or others, or at least it is available, and often they will be tracked. So can you extract the data freely from a universal database that will make it more accessible for others? You have an identifier, and therefore you can measure. Often these things measure how often is it downloaded. Where possible, you should use standard formats and vocabularies. You don't need to invent your own. You can use ones that exist already. To make it more reusable, we have rich metadata, and you apply community standards so that they can be re-examined as easily as possible. Obviously, that's a work in progress, but that's where we'd want to be. An example is the ISO 86001 date standard. Remarkably, um, people don't use this when it's using a date standard is pretty common for documents. It's nothing unusual, but you'll see all sorts of different date formats being used, which is quite bizarre. But the standard way is kind of year, month, date, and that's the way it should be maintained, and that makes it much more easy to track documents. Um, what else can we think about? So we're all familiar with structured data where you have a standard table with rows and columns. That's relatively easy to handle. We might have semi-structured data where we might have things like JSON files, XML files, data that has a structure. For example, we might have a set of people such as Emma here, who's a certain age and a certain birthday and a certain name. But compiling that information for a set of people may not necessarily mean that we can simply store it as structured data, maybe semi-structured. And then we can think about unstructured data where images, videos are examples of that. So we can think about the our analysis. So analyzing structured data is pretty easy, and often we want to take unstructured data and extract the features that are of interest and convert that into structured data depending on what we're trying to do. We can use what are called relational databases for structured data, which is easy, whereas for unstructured data, such as pictures on your phone, or semi-structured data, we may have another interface for looking at this. Um, so you can think, for example, for your unstructured data, if you think about pictures on your phone, often what's of interest is that you're identifying who was there and where was it taken and when was it taken. So that metadata is key, especially if you look at very old pictures. You might think, what year was that? Where was that? I don't remember. So having access then to information makes that image useful, and that's what we're talking about when we talk about data management. Semi-structured data lacks the rigid structure of structured data, and is typically dependent on the type of semi-structured it has. Often that fluxes during versions as it changes, and often the data includes tags or markers that allow it to be differentiated from each other. And JSON and XML files are examples of that. Um, If we look at unstructured data, there's no fixed format necessarily. You will have conventions within domains, but they'll not always be the same. We also have diverse data types. So text, images, audio, video, social media posts, many more things. There's many examples of this. They'll also be complex to process and therefore have their own particular methods for using that, and that are unique to that particular area, which can't be transferred easily to other unstructured data. So, anything like images, audio recordings would fall in there. For looking at biological data, images are the obvious one, but videos are also becoming more common. You might have analysis of papers to extract information from them, um, and you can see that each of these areas is quite different from each other. An example of this where we might have a raw photo that is transformed in some sort of way to make it more highly resolved, and then it has metadata linked to that. That's the key for making that above photo useful. So therefore, if it's stored in a standard database, we can access that metadata associated with that photo more easily. If it's not stored in the right way, then it makes it difficult to find that data to make it accessible and to make it more interoperable. So what we want is proper metadata and documentation. So if we have data linked with metadata linked to it, we want a separate spreadsheet for the metadata, describing its contents in detail, including the definitions of the column headers and so on. This is called a data dictionary. So you might have, for example, a set of variables, variable 123 here, and you might describe what type of variable there are, the binary, numerical, or the label. You might put in the units and the ranges of the data as well. We might have information on them saying that they're subspecies of infecting viruses. I might have a label that's associated with infection status. Another example here where we have a description of what's present, where we have the name is going to be a species. We have the plot name as um, mouse. The group is demographic and so on. So we've got these different labels on the left, and the detailed description on the right. So for example, the sex is going to be male or female in this case. We have coat color is comes from visual inspection. The crumblers depends if the mouse has stored food in their bedding, and so on. And using standard nomenclature is quite challenging for the community. So be simple, not sesquipedalian. You can see then if you look at microbiome studies where there's a lot of different terms for feces. So using the same term to describe something is probably actually a good idea in terms of making data more findable, consistent, and accessible for others. So you see all these different tools, different words that are used for feces, and this makes it more challenging to work with when you're trying to search for things. So for data, don't be creative, be accurate. Put one variable per column, which you can see here. We got sample one, sample two, sample three. You got the variable one, the binary classification. You got some sort of measure, and you got some sort of status here. You might have instances where you have multiple information in a column, which is not a good idea. So therefore, we can split this. And this is replicate one for the wild type, an infected replicate one, and an infected replicate two. So you can split them: wild type, infected, infected, and you got the replicate identifier there as well. If you need more columns, add them. There's no penalty on this. And most importantly, don't do calculations in spreadsheets containing your own data. Ideally, use computer code. How to analyze data faster. So you think about storing our data in one sheet. Don't split it across multiple sheets or files. Add a data dictionary describing the data that's inside the data. De-identify the data if you're using human data. Unmerge cells. You got one entry per cell only. Remove all blank rows or columns. Don't put them in your data. Don't put in calculations inside your spreadsheet. So avoid this where you got lots of extra columns and rows here. Um, these are all not good, and this is part of the data dictionary. It shouldn't be in the main spreadsheet itself. So missing values, typically for biology, it'll just be an NA. Um, and then if you've make sure your data is either a number or text. Avoid use numbers and avoid text as well. So if you have F and M here, might relabel it as one and zero. Um, use good naming. So have an informative name. So H category becomes H underscore cast. So if no space in there. If you have blood pressure, for example, here where you got 80 over 120, you can split them into two separate columns. And don't use hyphens because certain tools may mangle them. Another example is to code numerical. So if they're discrete values, then use those discrete values. And then avoid having asterisk indentation, colors, etc. to flag something. You can put in an extra column to flag that item. Format dates consistently. You can see this is not actually the ISO 8601 standard. You can split minutes, columns, hours, months, weeks, years, whatever. You don't need the units inside there. Um, so be consistent with whatever metrics you're using. Use informative short column headers without any symbols such as species, date, location. Um, and you can use identifiers to link data across related data across web sheets rather than duplicating data. If you don't do this and are inconsistent, you have different column headers and don't normalize the data. Um, I don't know what you're trying to do, but it's basically sabotaging your own project. So what's a good name? So max temperature C is a good name because you can kind of get the meaning from it there. Whereas this one, where it's all spelled out with the spaces and the symbols and the brackets, is not a good example. And there's lots of examples here. You can look at this paper yourself and see that there's some obvious things that should be done that will make your life more easy. What else? Spaces and decoding the data inside the column names is not a good idea. This should be in a separate data dictionary spreadsheet. What else can we do? We can remove duplicate entries, which can arise. We can add in NAs for missing values. We can have validation rules to check that um, the values are within the range that should be expected. Um, we never have spaces in our spreadsheet anywhere. Don't have blank rows or blank columns. Please also note that raw data is primary data. Process data has been changed. It's fine to have processed data, but it's not the same as the raw data. So the best way of thinking about it is like cooking. It's irreversible. So if you convert your chicken along this pathway, it's very, very difficult to go back the other way. So think of it that way. Once you start changing your data, it's hard to go back. So you can have separate analysis data sets where you've analyzed the data, but that's not the same as your raw data. So you might have your raw data, your clean data, and then from that you might have multiple different analyses that are completed, and you may have spreadsheets that are for visualization or tables that are for visualization, but they're not the same as the cleaned data. They're separate. So these are the simple steps. Create a data dictionary, create a detailed data cleaning planning using the data dictionary, and what does your community do? Import the dataset into R, and then you can visually export there and do changes with code rather than doing it manually. So key other things to making your data machine readable. So that's really what we want to go. We're not thinking about human readable, but machine readable. Avoid merged cells. Do not put symbols in your dataset. Don't use colors. Add an extra column to indicate whatever you want to indicate. Use consistent data types. Use standard codes. So one column is one variable, one row is one sample, one cell is one piece of information. The first row is the header, and the header names do not have special characters like spaces. So example of this is like this, where the strain is A or B, the particular genotype, either normal or mutant. The minimum, this could be minimum value that's observed, and then we've got the replicative column, and they've got the response column. So that would be a good way of organizing the data. It's very, very simple and easy to read with, and you'd have the data dictionary associated with it. So that kind of concludes our initial video then on looking at um, making your data AI ready. Please see our next video on types of AI methods.